In today's world, drugs have become a societal problem. Drug designers are now making deadly recreational drugs such as methamphetamine through over-the-counter medicines. One such medicine is a common nasal decongestant, Sudafed.
The active ingredient in Sudafed is pseudoephedrine, hence the name Sudafed. But how is this drug a miracle nasal decongestant as well as a potential illicit drug? To understand this, we must first understand how the structure of pseudoephedrine functions in our body. Pseudoephedrine has a secondary amine, an alcohol, and an aromatic ring. This relatively simple structure of pseudoephedrine has a measurable value as a nasal decongestant that relieves pain, pressure, and congestion.
Although the mechanism of action of pseudoephedrine in the human body is still undergoing research, we know that it aids in the release of a neurotransmitter known as norepinephrine. Some say that pseudoephedrine has weak or no direct agonist activity with any of the adrenergic receptors. Others say that pseudoephedrine acts directly on alpha and to a less degree beta adrenergic receptors. These receptors are GPCRs, meaning they release norepinephrine through a signal transduction cascade. As we have learned in Chem 332, we can see that these two structures look fairly similar. Pseudoephedrine is known as a sympathomimetic drug, which means that it mimics the effects of the sympathetic nervous system, i.e. norepinephrine. So there is a potential of molecular mimicry taking place, allowing the adrenergic receptors to recognize both pseudoephedrine and norepinephrine. It's norepinephrine's binding to alpha adrenergic receptors that results in vasoconstriction and decreased mucus production, while relaxation of bronchial smooth muscle is stimulated through its binding to beta adrenergic receptors.
Here we see an example of a GPCR, the beta adrenergic receptor, and its 7 transmembrane domains. When a ligand binds to the GPCR, it causes a conformational change, which can then activate an associated G protein by exchanging its bound GDP for a GTP. This is critical for initiating the downstream signal transduction pathway that will eventually release norepinephrine.
Current research on these adrenergic receptors deals with the properties of GPCRs known as ionic locks and rotamer toggle, both of which are means of activation of the GPCRs. Recent fluorescence spectroscopic studies by Yao et al. Show the ionic lock between transmembrane 3 and 6 in these adrenergic receptors. Here we see a JMOL image of a beta adrenergic receptor. With this structure, we can see that it has an alpha helical domain with a single chain. By rotating this receptor, we can recognize the active site, and by further rotation, we can recognize the characteristic beta sheet of the GPCR. This JMOL image is of the alpha 2 adrenergic receptor. Again, this has an alpha helical domain with a single chain. You can see the distinction between the beta and the alpha adrenergic receptors through this beta sheet. In this expanded view of the receptor rhodopsin, we can see the toggle switch and the ionic lock location. The pink structures are the rhodopsin residues and the blue structures are the general adrenogenic receptor residues. The superposition or the overlapping of the aromatic residue cluster involved in the toggle switch emphasizes the activation of the enzyme.
So how exactly is meth made from pseudoephedrine? This process is known as the Birch reductions. The conditions of this reaction are highly basic due to ammonia and highly reactive due to the radical formation. This is the main reason why deadly explosions occur at home meth labs.
To start off the Birch reduction, it requires an alkali metal in solution, usually lithium. This lithium will cause the formation of a carbon radical, which will then push these pi electrons to form a carb anion. After the formation of the carbon radical and carb anion, the molecule will undergo a beta elimination in which the carb anion will push its electrons to form a double bond, causing the alcohol group to leave. Then another lithium molecule will donate an electron to form a carbon ion, which will subsequently undergo a beta elimination followed by an association of electrophile step. Through these steps, we will form our final product of methamphetamine. Here we can see the HOMO and LUMO interactions taking place in the Birch reduction mentioned before. Zooming in on box 2, we can see the sigma star orbital between the oxygen and the carbon indicating its removal. Zooming in on box 4, in the HOMO we can see this N to pi star activity indicating the formation of a double bond, followed by the protonation of a carbon, giving the structure of methamphetamine. What are the different effects of pseudoephedrine versus methamphetamine? Pseudoephedrine is known to cause vasoconstriction, decreased mucus production, and relaxation of bronchial smooth muscles. Methamphetamine is known to increase alertness and energy, but in high doses can lead to excessive usage due to induction of euphoria, enhanced self-esteem, and increased sexual pleasure. In conclusion, the molecule of pseudoephedrine truly shows the power of chemistry and how simply one reaction, like the Birch reduction, can change the body's entire response from a miracle decongestant to a potential illicit drug.